Hello everyone and welcome to Instant Biology by Dr. Neelab. I am here with another very important lecture that is important with respect to the examination, competitive examination as well as the university level examination. The topic that I have chosen today is FRAP, that is also called as fluorescence recovery after photo bleaching. Now this is a technique that is often used in cell biology in order to study the movement of the lipids and the proteins. So in this technique I would be talking about multiple things. I would be talking about what is the principle of this technique. I would be talking about how this technique is used or executed. I would also be talking about the different questions that have been asked in CSIR net. I'll be showing you some very useful GIFs, some very useful diagrammatic representations. So this would be a wholesome lecture. I have already made a lecture on FRAP in Hindi. So those of you who are interested in that lecture can get the link over here, and you can watch that lecture, and this would be in English, completely in English. So without much ado, let us start. What is FRAP? Fluorescence recovery after photo bleaching. What is this technique? What is the purpose of this technique? Remember, whenever you are studying a new technique, you should first of all put your brains into finding out why this technique is used. Yeah, what is the purpose of this technique? Because until and unless you understand the purpose of that technique, you would not be able to understand the principle or mechanism. So what is the purpose of this technique? Lateral movement of specific plasma membrane proteins and lipids can be quantified by FRAP. Now let us understand this one by one. What I want you to understand is lateral movement. Now this is a word that might be new to you. Lateral movement, what does this lateral movement mean? Lateral movement means movement in the same leaflet. Let us just reconsider the definition first of all. Lateral movement of specific plasma membrane proteins and lipids can be quantified by FRAP. So you can say that the movement of the plasma membrane and proteins can be quantified, it can be detected through FRAP. What do you mean by plasma membrane, lipid and protein? Let us first of all understand this. You might be knowing that plasma membranes are made up of two things, majorly two things. They are made up of lipids and they are made up of proteins. So these are the two things that plasma membranes are made up of. Okay, now having understood this, you should be knowing that this is a diagrammatic representation of your plasma membrane. However, proteins have not been shown over here, only phospholipids have been shown over here. This, you can consider that this is a phospholipid, this also is a phospholipid. You would also know that plasma membrane is made up of two layers. It is basically it is having two leaflets, we should say. Now this is the upper leaflet. Let me just rub this out. So this is, let us consider this as the upper leaflet, and this is the lower leaflet. So we are saying that plasma membrane is made up of two leaflets, upper leaflet and lower leaflet. Okay, so both of these are made up of phospholipids. So when I say that the purpose of this FRAP technique is used to detect the movement of the phospholipids in a lateral way, then we mean that suppose this is a phospholipid, the one that is stained in the red color. So suppose this is a phospholipid and I have numbered them 1, 2 and 3. So initially it is in the third position. This phospholipid is in the third position. What I want you to understand is that the nature of the plasma membrane is dynamic. That means that continuously there is a movement of the lipid as well as the protein molecules inside the plasma membrane. So if this happens, understand this, the lateral movement means when this phospholipid in this leaflet is moving from the third position to, let us say, somewhere over here. Over here you can see this diagrammatic representation. So previously this phospholipid was somewhere over here, but now it changes its position and is now at number 8. So you can consider, you can just number it 1, 2, 1, 2, 3, 4, 5, 6, 7th and 8th. So now previously the phospholipid that was at number 3 is now at number 8. How can you detect it? Let's say that it was stained over here, red colored stained, and the stained phospholipid has moved on to the 8th number. Remember, when I am saying lateral movement simply means that movement in the same leaflet. I am not saying that the movement will be from here to here. Lateral simply means that movement in the same leaflet. If it is in the upper leaflet, it is moving in the upper leaflet only. If it is in the lower leaflet, it is moving in the lower leaflet only. I am not saying that flipping and flopping cannot take place, or a phospholipid cannot jump from a upper leaflet to a lower leaflet. I'm not saying that. That can also be possible, and the process is called as flipping and flopping. But as of now, I am only talking about the lateral movement. So this is the example of lateral movement. This type of movement can be detected by FRAP. It can be quantified by FRAP. So this is basically the purpose of this technique. I believe you would have understood this. So this figure is depicting the lateral movement of phospholipids. I believe you would have got a clear idea about this figure. Now, coming to the lower point, rate at which the membrane molecules move can be identified with, it is also referred to as diffusion coefficient. What I want you to understand over here is the rate or the velocity at which these molecules are moving, these lipid molecules or the protein molecules are moving in the plasma membrane. That can be quantified, that can be estimated, that can be determined, and basically it is referred to as diffusion coefficient. What do I mean by diffusion coefficient? By diffusion coefficient, I simply mean that the component that has a higher diffusion coefficient will be moving at a faster rate as compared to the molecule that is having a lower diffusion coefficient. Okay, so again, saying this again, the molecules that are having higher diffusion coefficient will show a higher mobility as compared to the molecules that are having lower diffusion coefficient. So this is estimated by the fluorescence recovery after photobleaching. It will basically give you a diffusion coefficient through which you can estimate, through which you can get an idea that which component is moving faster, which component is moving slowly. So this is about the first diagram. I hope you would have a clear-cut idea about this diagrammatic representation. Now let us move on to the next slide. Yes. So also the proportion of the molecules that are laterally mobile are also identified. What proportion of molecules? What do I want to say when I say proportion of molecules? See, I'm not saying that in a plasma membrane all the molecules are mobile. I'm not saying that. There are certain components that are not mobile. Of course, there are certain components that are mobile and there are certain components that are not mobile. So the proportion of the molecules that are laterally mobile, it will give you an idea what is the proportion of the molecules that are mobile. What is the proportion of the molecules with respect to the non-motile molecules? What is the proportion of the motile molecules with respect to the non-mobile molecules or non-motile molecules? This can also be identified with respect to or with the help of fluorescence recovery after photobleaching, FRAP technique. Now, what is the method or how this is done? So you would have understood the purpose, you would have understood a good idea, you would have got about why this technique is done or what is the purpose of this technique. Now coming down to the methodology, how this technique is executed. First thing, step number 1, now you have your cells. Now you label the cells with a fluorescent reagent. What you have done is you have labeled the cells with a fluorescent reagent. Now I want you to understand over here very clearly, when I say cells, I am talking about the plasma membrane over here. So the plasma membrane, when I am talking about the cells, I am referring to the plasma membrane over here, because we are concerned with the plasma membrane only, because we are talking about the components of the plasma membrane only. And what are the components of the plasma membrane? You already know that the components are lipids and proteins. These are the major components. Apart from lipids and proteins, there are carbohydrates also, but they are present in a very, very low concentration. So I'm not going to discuss about that. I'm talking about the lipids and the proteins. Right. So the cells or the plasma membrane of the cell is made up of lipids and proteins. Now lipids are basically termed as phospholipids. Why do you call it as a phospholipid? Because of the attached phosphate group onto the lipid moiety. Right. Next, Phospholipid containing fluorescent substituents. What I want you to understand over here is, I have already said that you have to label your cells with a fluorescent reagent. When I say fluorescent reagent, I mean that when you are hitting the molecule with a laser light, or when you are hitting the molecule with a light of a particular wavelength, what will happen? This molecule, because it is fluorescent molecule, it will also emit a light of a different wavelength, and that light can be detected. Okay, that is called as fluorescence. So you can detect that light. So what I have done is, I have stained the cells, I have stained the lipids or proteins, whatever I want to detect, I am staining that with the fluorescent dye. So that is what is written over here: phospholipids containing fluorescent substituents. If you want to stain the lipids. If I am talking about proteins, in case of proteins, map specific exoplasmic domains of desired protein tagged by a fluorescent dye. Simply you can understand that monoclonal antibodies are used. In case if you want to label a protein, monoclonal antibodies are used, and these monoclonal antibodies should be specific for exoplasmic domain. What I want you to say is, exoplasmic means non-cytosolic domain. These monoclonal antibodies should be binding to the non-cytosolic domain, and these antibodies should be tagged with a fluorescent dye. Okay, so over here you can see that this is suppose a protein. Over here you can see that this is a protein, and this protein is bounded by this antibody, and this antibody is connected with a fluorescently labeled tag. This is what I want to say. Again, this is suppose if this is the protein, I have to have an antibody. This is my antibody. Let us say this pen is an antibody. This pen would be binding to this protein, and this is, let us say, fluorescently labeled molecule. This is binding to this antibody. So this antibody is already binded to this fluorescently tagged molecule, and then fluorescent molecule, and then it is binding to the protein. So automatically what will happen? This protein will be tagged in directly. This is what I want to say. So this is how you would be fluorescently labeled. You will be fluorescently you will be able to fluorescently label the protein as well as the lipid molecules. Whatever you want, you would be able to do that. Now let us move forward and discuss about the about how this labeling is done. So this is a fairly good diagram. You can check this out. So suppose these are the membrane proteins initially. You can see that there is a particular stain over here. These are not actually stained. I'm just showing it to you that this is the normal cell membrane, unstained cell membrane, unlabeled cell membrane rather. Now when you next label it, what are you labeling it with? You are labeling it with fluorescent dye. So this fluorescent reagent stains the entire membrane. You can see that this is all the fluorescent labeled the phospholipids that are all fluorescently labeled. Now when we move ahead, what I am talking about this is, you are bleaching this this phospholipid layer, you are bleaching it with the help of a laser light. What will happen when you are using the laser and laser hits the phospholipids that are fluorescently labeled? These phospholipids will lose their fluorescent labeling. Just over here you can see that this is the bleached area, and in the bleached area you can see that there is no fluorescent labeling, right? Next we are talking about fluorescence recovery. I have already said that the nature of the plasma membrane is mobile or dynamic. That means the components of the plasma membrane would be continuously moving. So this is a fairly good example that after some time, after you have bleached the plasma membrane, after some time you will see fluorescence recovery taking place. What does that mean? It simply means that the unlabeled phospholipids would have moved from their exact position to the different other areas. So you can see over here, previously the bleached molecules or the bleached phospholipids were located in this area, but now they have moved or they have shifted places, they have changed their place like this. And also the labeled molecules, the one that were previously labeled, have also moved, and they are also now present in the previously we were calling it bleached area. Now in the bleached area also you can see these are the labeled molecules. Okay, so this entire experiment suggests that When you are initially labeling the phospholipids, all the phospholipids will be having that particular stain, and once you are hitting it with the laser light, because of the laser light the fluorescence will be lost, and then after some time fluorescence recovery will take place. Fluorescence recovery simply means that the bleached area, the area that was previously not emitting out any fluorescence, would now start emitting out fluorescence, because the labeled molecules have started moving in that area and the unlabeled molecules are also moving in some other places. So there will be a mixture of the labeled and unlabeled molecules in the last diagram. You can see that. Now same thing is being depicted over here. So this was the A diagram and this is the B diagram. What I want you to understand over here is, now initially this is on the y-axis we are showing fluorescence intensity, and on the x-axis we are showing time. Understand this, that once you are bleaching, the bleaching point is over here. Once you are bleaching, what is happening? So this was the initial fluorescence level. This was the initial fluorescence before bleaching, somewhere around 25 or 2800. Then after that, once you did the bleaching, suddenly the fluorescence came down. It is up till here. This is the level of fluorescence. It suddenly came down. This is the point that I am talking about. Because you have bleached, this area is not emitting any fluorescence, so it came down suddenly. Previously, let us say it was this much, that it suddenly came down. Now again, this is the process of fluorescence recovery. Again, after some time it will start recovering, something like this. So this fluorescence recovery is taking place. Now how much will be recovered? If I talk about what would be the recovery and how much it would be recovered, the recovery would be depending upon the amount of the mobile components present. More are the mobile components, more faster will be the recovery. So over here you can see that if I say that 50% recovery has taken place, if I say that 50% recovery has taken place, so this is the amount of recovery that has taken place, because the graph again rose from here till here. So this is the amount of recovery, but this portion was not recovered. So the portion that was not recovered is because of the immobile components. The portion that was not recovered was because of, let me just rub it this. So the portion that was not recovered, rather I should rub this also. Yes. Yeah. So this portion is due to the immobile components. Immobile. And this portion is because of the mobile components. Right? So the recovery portion, the portion that has recovered is due to the mobile components. The portion that was not recovered is because of the immobile components. Right? We could not recover it completely, because let us say that the level over here is around 2000, but the initial level was somewhere around 2800. So we were not able to recover it fully. So the unrecovered portion is because of the immobile components, the recovered portion is because of the mobile components. So I believe you would have understood this. Now let us move on to the next slide. So in the next slide also I am showing you the same thing, but with a different orientation. You can see that, suppose this is a side view of a cell, and the cell is, you can see that the cell is stained with, let us say, red fluorescence dye. So the side view, you can say that this basically all the phospholipids over here are stained with red fluorescence dye. Okay, now what we do is, suddenly we use laser. When you are using laser, this is the side view, remember, certain portion of this phospholipids will be losing their fluorescence. They have now become black. That means they have lost their fluorescence. When you convert this into the top view, if you are supposed to observing from the top, then what will happen? This portion is referring to the phospholipids that have lost their fluorescence because of the laser. Now, as I have said that as the time would progress gradually, you would see that the plasma membrane would show fluorescence recovery. That means the lipids, phospholipids from nearby regions will be moving, and the phospholipids that have lost the fluorescence would also be moving. So gradually you will see a mixing over here. After some, initially you would see a mixing that has started, and after some time you will see almost complete mixing. So over here you can see that this black colored region, this bleached region, has started to become normal. Initially it was very prominent, but now it has become a little dull, because again the fluorescence recovery has started over here, and over here fluorescence recovery has completed because the black region has already been lost. Yes, of course, I'm not sure if you are able to distinguish between these two areas, but this is slightly darker, this is slightly lighter, obviously because of the black regions present over here, or because the complete fluorescence recovery would not have taken place, only a percentage of recovery would have taken place. That is why this region would be comparatively darker. This would be comparatively darker as compared to this one, or for the matter of fact this one, rather we should say that this is slightly darker and this is slightly lighter, because over here fluorescent recovery is not 100%. There is some percentage of recovery, because not every component inside the plasma membrane would be mobile, some would be immobile. Because of that only the fluorescence recovery has not completely taken place. This is what I wanted to show you. In this diagram again you can see that initially the level of fluorescence was this much, suddenly you applied bleaching, because of the bleaching the level of fluorescence went down, and again because of the fluorescence recovery, this is fluorescence recovery, and because of the recovery the fluorescence again reaches to this area. The recovery has been done because FM, this means fluorescence recovery because of mobile components, fluorescence recovery because of fluorescence. This region was not recovered because of immobile components. So this is what I wanted to show you in this particular diagram. I hope this would have made sense to you. Now let us move on to another diagrammatic representation. The same way you are seeing this. So you perform the photo bleaching. In this diagram you can see that you perform the photo bleaching. Because of the photo bleaching, this area turned this area was not fluorescing, and this again gradually the recovery took place. The same diagrammatic representation is present over here, and this recovery is being depicted by diffusion coefficient that I said. The fluorescence recovery after photo bleaching gives you actually the diffusion coefficient value, and with the diffusion coefficient only you would be able to realize that how much is the mobile component and how much is the immobile component. Okay, now let us move to the next point. Now step 2, I have already discussed all of these steps through the diagrammatic representation, so let us quickly move through them. So step 2, laser light was focused on a small area and it caused bleaching to that area. Fluorescence was reduced in the illuminated area. Step number 3, after some time fluorescence can be seen in the bleached patch because recovery started happening. Explanation: extent of the fluorescence recovery in the bleached patch was directly proportional to the fraction of labeled mobile molecules. So this point, this explanation, I have already said that the recovery of the fluorescence recovery would be directly proportional to the number of components that have the mobile nature. Okay, so this is what is mentioned over here. The fraction of the labeled molecules or mobile molecules would be actually determining that how much fluorescence recovery would be there. Now next, human hepatoma cells of asialoglycoprotein, basically 50% fluorescence returned, and because this shows that if 50% fluorescence was returned in this particular protein, then we can assume that 50% mobile nature was there. Okay, having said that, let us move on to the next point. So what components are mobile and what components would be moving more and what components would be moving less? Let us talk about this. So if I am saying that amongst the components of the plasma membrane, the lipids would be moving the highest, or lipids would be showing the highest mobility. Okay, proteins would also be mobile, but lipids would be the most mobile components. So this is the phospholipid. It will be showing the highest mobility. Next, single pass transmembrane protein. Single pass transmembrane protein means when the protein has crossed the plasma membrane only one time. So you can see that only once the plasma membrane has been crossed, so therefore it is also called as single pass transmembrane protein. So just after lipid, the next mobile component would be single pass transmembrane protein, followed by this multi-pass transmembrane protein. So multi-pass means it will be crossing the plasma membrane multiple times. This is called as multi-pass transmembrane protein. Finally, multi-pass transmembrane protein plus cytoskeleton. That means this is a multi-pass transmembrane protein and it is also joined to cytoskeleton molecules. You might be knowing what are cytoskeleton molecules. They can be microtubules, they can be actin filaments, they can be intermediate filaments. So all these components can be present over here. Obviously, this is very logical. Lipids would be having the highest mobility. Single pass transmembrane protein, because it is not connected with any other thing, plus it is not showing multiple passes. Obviously, it will be comparatively free to move, so it will be the next one to be mobile. The third one would be multi-pass transmembrane protein. It is crossing the plasma membrane multiple times, so it has reduced mobility. But the final one, or the least mobile one, would be the multi-pass transmembrane proteins, which are connected with the cytoskeletons, right? So this is because they are crossing the plasma membrane many times, they are attached to the plasma membrane more highly, and furthermore they are also joined to the cytoskeleton molecules. So this is the entire thing. I believe you would have understood this thing. Now this is a diagrammatic representation. You can see that initially, again when it will play, so you can see that all of them are green in color initially. All these plasma membranes, all these phospholipids are green in color initially, but then you can see that this is the laser flashing. As the laser flashes, this fluorescently labeled phospholipid molecules, they will turn into non-fluorescent molecules, because you can see that these are the black ones, these are non-fluorescent, and automatically they will start moving because of the mobile nature or because of the dynamic nature of the plasma membrane. So again and again you can see the same thing happening. So this is a GIF with which you can easily understand how this works. Now let us move on to the questions that have been asked from FRAP in CSIR-NET. So first question, using FRAP, fluorescence recovery after photobleaching techniques, diffusion coefficients of three integral membrane proteins M1, M2 and M3 in a kidney cell is calculated. 1 μm per second, 0.05 μm per second and 0.005 μm per second. Considering fluid mosaic nature of biological membrane and relationship of structural organization of integral membrane protein with diffusion coefficient, which protein will have highest number of integral membrane domain? So let me just rub this. It's not required, it's not rubbing. Let us see that why the correct answer is 3, but I would want to explain to you that why is the correct answer 3 basically. As the coefficients, diffusion coefficients have been given for the three proteins M1, M2 and M3. So in case of M1, the value has been given as 1 microliter per second, 0.05 μm per second and 0.005 μm per second. These are the three values that have been given to you. Understand this. I have already told you the one which will be having the highest coefficient will be the most mobile. So it is basically saying that which proteins will have highest number of integral membrane domains. So obviously the one which is having the least diffusion coefficient will be the least mobile, and if it is least mobile, obviously it would be more connected, it will have a multi-pass nature, obviously it might also be connected with the cytoskeletons. So obviously because this one M3 has the least coefficient, we can understand that it would be having a high number of passes, plus it might also be connected with cytoskeleton proteins. So with this we come to the conclusion M2 or M3. Basically M3 has the lowest ones, so we can say that M3 only is the correct answer, or you can say that it will be having the highest number of integral membrane domains. Okay, so with this we complete the CSIR December 2013 question. Now let us move on to the next question. So this is another question that has been asked in CSIR June 2016. Lateral diffusion of proteins in membranes can be followed and diffusion rate calculated by. So with which of these techniques you can calculate the diffusion rate? Obviously we have already studied this. Diffusion rate will be calculated by FRAP, fluorescence recovery after photobleaching, fourth point. Okay, now let us move on to the next one. So this is a very important question. Fluorescence recovery after photobleaching, FRAP, is a method to estimate the diffusion of molecule. This is about FRAP. Fluorescence labeled molecules such as a receptor tagged with green fluorescent protein, a receptor labeled with green fluorescence protein which interacts with cytoskeleton, a labeled lipid, a labeled protein that binds to the membrane surfaces are photobleached, and the recovery profiles A to D were obtained to estimate their diffusion coefficients. Now they are asking, basically following data was obtained when these are the different options, these are the different things that was stained by GFP and then they were bleached. What would be the recovery profile they are asking? So obviously I have told you, if you see over here, a lipid protein that binds to a labeled protein that binds to the membrane surface, and there is a labeled lipid. I have already told you that the lipid that is labeled will show highest fluorescence recovery. So the highest fluorescence recovery is being shown by B over here. You can see that this is showing the highest fluorescence recovery. So B can be labeled with, B can be joined with 3. B can be connected with 3. Next, a receptor tagged with green fluorescent protein, a receptor labeled with GFP which interacts with cytoskeleton, a labeled protein that binds to the membrane surface. Now you know that a labeled protein that binds to the membrane surface, basically it is binding to the membrane surface. It is not basically showing the integral nature. It is not showing the integral membrane protein. It is not crossing the membrane multiple times. It is just a surface protein, or you can say it is a peripheral protein, obviously. So what would be the case? The next one would be the peripheral protein that we are talking about. The one that is peripheral, it is not showing the integral membrane nature. Others, receptor tagged with green fluorescence protein, a receptor would be an integral membrane protein. Again a receptor, this is again a receptor would be integral membrane protein, plus it is also binding with the cytoskeleton. That means it would be more hindered, it will be more limited to move. So again we can label that the fourth one would be, can be joined with the next fluorescence recovery graph. So this A is just after B. That means it will slightly be lesser, it will slightly show lesser recovery than B. So we can say that the fourth option matches correctly with A. So I believe B3 and A4 would be the correct option. So there is only one options like this. So B3 and A4 would be the correct option over here. Others also you can see, basically this would be the third last one, I believe. So if you can see that this D shows next recovery. So D, I believe, would be the first one, and the last one would be 2. C can be joined with 2. This would be the final answer. Okay, so I have arranged all of them according to whatever I have taught you. Now let us move on to the next point. Next question. So in this question you can see that this has been asked in CSIR 2019. To investigate the dynamics nature of two unrelated centrosome localized GFP tagged proteins, GFP-A and GFP-B. So there are two proteins, GFP-A and GFP-B. A team of scientists conducted fluorescence recovery after photobleaching experiment. The FRAP profiles of these two proteins have been given below. So these are the FRAP profiles. Now you have to check out which statement is correct. The following statements for this FRAP analysis were made. GFP-B shows faster exchange rate than GFP-A. So GFP-B shows faster exchange rates. Of course, what do you mean by exchange rates? Exchange rate means fluorescence recovery. So obviously GFP-B has a faster fluorescence recovery as GFP-A. This is obviously shown by the graph. So I think this option A is correct. GFP-A shows faster exchange rate than GFP-B. Oh, sorry. GFP-B. Obviously, GFP-B is showing faster exchange rate than GFP-A. The second option shows GFP-A shows faster exchange rate than GFP-B. No, GFP-A is lower than GFP-B, so it is showing lower exchange rate. So this option does not seem to be correct. GFP-A has more immobile fraction. GFP-A has more immobile fraction than GFP-B. Obviously, it is having more immobile fraction than GFP-B because GFP-B is having only this much of immobile fraction, but GFP-A is having this much of immobile fraction. So obviously there is a difference, and GFP-A is having a higher immobile fraction. So I believe GFP-A has more immobile fraction. This also is correct. GFP-B has more immobile fraction. This is wrong. I just told you the reason. So what are the correct options? A and C. So A and C are present over here. That means the first option is correct. Okay, so this was the next last question that I wanted to take. I believe you would have a clear understanding about the concept of the fluorescence recovery after photo bleaching, and you would have understood each and everything, and also the questions that I had asked. One more important thing that I wanted to tell you is that we have started our bioprocess engineering second batch, and this batch is have started a couple of days back, and the class starts at 6 p.m. in the evening, and it runs for an hour. So 7 p.m. it ends. Monday to Saturday it will take place, and we will be completing the course in 1 month for bioprocess engineering unit number 4 and unit number 5 for gate examinations. If you are interested to know more, obviously you would get the link above over here, and if you want to contact us, you can contact us on this number 8299134528. So this is our contact number, 8299134528. This is our contact number. You can also get this details. You can also mail us. Everything is given in the description section. You can get in touch with us. So I hope you would have enjoyed this lecture. Thank you so much. Have a good day.